Hi, everybody, and welcome to the afternoon session. I'm David Goldstein. It's my pleasure to introduce the first speaker in this session, Gale Hammell from Cold Spring Harbor, talking about mobile genomics.
Thank you to the organizers for inviting me to come give a talk. It's a pleasure. I've never given a talk here in the Genome Center auditorium before, so this is the first for me. It's quite fun. Yes, so I'm going to be telling you a little bit about transposon biology and how we've been able to link that. We have supportive evidence to link that to the neurodegenerative disease ALS, but I'll take you through that bit by bit. So I just want to remind you what transposon sequences or mobile elements or transposable elements are. These are mobile elements that at least at some time in the past have had the ability to hop in the genome. So there are a very, very small number of these that still have that ability, that are competent to hop, and when they do, the retrotransposons, which is a subset of them, will basically make an RNA copy and then reinsert themselves into a new locus in the genome. Okay, and so it turns out that they've been very successful over evolutionary timescales, such that they're about half your genome, right? So most of those are made up of this retrotransposon subset that I was telling you about. They go through an RNA intermediate. Some of these retrotransposons are actually proper endogenous retroviruses, but that's actually a small fraction of all of the retrotransposons. And then a very, very small fraction of the human genome is derived from DNA transposons, and these are the subset of transposons that Barbara McClintock originally discovered in maize when she was trying to understand coloration patterns in maize kernels, right? So I think we always have to pay a little bit of honor to Barbara because she figured out that these things were mobile in the genome long before we knew what a gene looked like or what a genome looked like or the structure of any of this. So I think it's kind of amazing. Things are a lot easier now. So even if you don't care about transposon biology, you think this is kind of a fringe, junk DNA kind of thing, you probably should still care at least a little bit. And my reasons for that are basically that they actually are in your sequencing reads, right? So they're about 5 to 10% of a typical RNA-seq data set, regardless of whether you're hitting one of the transposon silencing complexes or not. They're 20 to 50% of ChIP-seq data, depending on whether you're hitting one of these things that's enriched in heterochromatin or not, which is where a lot of the transposons lie. And they can be a very large fraction of small RNA data sets as well, right? So the idea that they can hop, right, is not just something that's completely irrelevant to human disease. This has actually been discovered that de novo insertions of active transposon sequences have been linked to specific human diseases. So this is an example from hemophilia, where line 1, the most fully competent active transposon in the human genome, actually jumped into the hemoglobin locus and caused hemophilia in this patient, right? But that's not the only way that transposons can be dangerous, right? Some of them will actually hop in and disrupt gene function in the way that I was just telling you about, but there's lots of other things that can happen when transposons are desilenced and when they're active. So one is that they can alter gene regulation of nearby genes. They can spread silencing marks, spread chromatin marks across to nearby genes and affect the regulation of those genes that way, and I'll give you a little bit more evidence later. So the idea is they don't have to actually be hopping in order to potentially be causing trouble, right? Okay, so things are sounding pretty grim, right? Transposons, they're everywhere in all plant and animal genomes. In some species, they actually outnumber host genes. In maize, it's 80% of the genome that's actually derived from transposon sequences, and they hop around making copies of themselves, sometimes destroying genes and certainly accumulating into large fractions of your genome. So why don't you hear about these all the time, and why are we mostly okay most of the time? And there's a couple of answers, but probably the biggest one is this. Most of those transposons that I was just telling you about that make up half your genome are essentially fossilized remains, right? These things have accumulated mutations so that they're no longer functional, they're no longer competent to jump, and they're no longer harmful enough to be completely deleted, right? Our genome hasn't completely gotten rid of these things, and they may or may not have been fully recycled yet. So some of them are recycled exapted, such that pieces of the transposons become pieces of coding genes or pieces of cis-regulatory sequences. But for most of them, they're essentially fossils. Okay, and in addition, for the few that are harmful, so for that small fraction of 1% left that are still active and competent and can hop, your cells actually devote enormous resources to preventing transposon activity, especially in germline cells where that can be passed to the next generation, right? So at every step of the transposon biogenesis pathway, there are complexes in the cell that are designed to silence these things, right? So that's everything from DNA methylation or transcription factors that repress transcription of these things, small RNAs that will digest the transcripts, and I'll talk a little bit more about that later, surveillance proteins that go around looking for transposon transcripts or transposon proteins generally in the cell, right? And so all of these different pathways together mostly keep these things silent most of the time. Okay, so, and the last question that I had is, why don't you hear about this more often? Why don't you hear about more diseases that are due to dysfunctions in transposon silencing or escape of these things? And while many excellent labs have been working on transposon biology for years, only recently have we really had better tools to actually look at these things, right? Generally, now that we have longer reads with fewer errors, we can actually consider these in genome sequencing data sets or transcriptome sequencing data sets, when before that was really quite difficult. In addition, we're developing better software that actually does statistical read assignments. So if you have 10,000 or 1 million copies, which is not uncommon for one of these line or ALU elements, and you're getting little pieces of them and trying to put them back and guess exactly where they came from in the genome, statistically that can be a very difficult problem. And so one of my postdocs, Ying, actually works on better statistical algorithms to actually statistically and probabilistically assign those reads to the most likely source locus. And things like that are helping us so that someone who hasn't been in the transposon field for 10 or 20 years can come and just say, hey, I think my protein might have something to do with this. Can we just look and see, right? And so that's one of the things that we do in my lab, is make software for that. But the other thing that you can do is you can use what you know about how transposons are controlled, which complexes are involved, which genes are involved, to try and predict diseases, human diseases that might actually be related to transposon activity. And this here is a picture of Lou Gehrig, so you can probably figure out where I'm going with this. So like I said, we're using these new tools and these better sequencing strategies that we have to go find new regulators in the transposon control pathway, and we're finding new ones all the time that you might not have thought of as a typical transposon silencing complex gene, but it actually turns out to have a role either predominantly or moonlighting as part of the transposon control pathway. So we're finding new regulators all the time using these new tools. And the latest of these is actually a protein called TDP43. So what is TDP43? So TDP43 is one of these aggregation-prone proteins. It's very likely, it has a prion-rich domain at the very end that makes it very likely to aggregate. Almost all ALS patients actually have aggregates of TDP43 in the cytoplasm. This is a hallmark for that disease, kind of like amyloid beta or tau proteins in other diseases like Alzheimer's, but this is the one for ALS, right? And so for a long time, people were trying to figure out, they knew these aggregates were here, but they wondered, is it the aggregates that are the problem in this disease, or is it loss of the normal function of TDP43? Because TDP43 is normally here in the nucleus, playing a lot of roles in mRNA processing, and when it forms these aggregates, and even prior in this pre-inclusion stage, it basically goes out to the cytoplasm and it's no longer forming these nuclear roles, right? So one question would be, what are its normal roles, and is that something that we need to really worry about when we're looking at these patients with TDP43 inclusions in terms of how to recover that function? So, okay, it clumps in a very large fraction of ALS patients. So this is Lou Gehrig's disease here. It's actually present, TDP43 aggregates are present in a range of neurodegenerative diseases. So it seems like even maybe 30% of Alzheimer's patients show TDP43 aggregates in the cells in addition to the other proteins that are typically there. Okay, so why am I telling you all this? Why do we think that TDP43 interacts with transposons? Well, TDP43 is an RNA-binding protein. You can immunoprecipitate this out of cells, and you can ask, what are its normal targets? And we kind of had a hint from some genetic evidence earlier that this was actually going to perhaps be interacting with transposon biology, which is why we really went after this. So we pulled it out of cells. We asked what its targets were. And this is all work that was started in collaboration with Josh Dubnow, an excellent collaborator of mine who is at Stony Brook now, and done by my postdoc, Nick. the ClipSeq worked, so we got the known binding sites for TDP43 and FUS. That's sort of one of its known targets, right? I can tell you that that worked. And in addition to that, it turns out that TDP43 binds transposons, and it binds lots of them. And this is really not typical for your average RNA binding protein, right? So here are the ClipSeq reads here piling up on these line 1 elements as they're dispersed throughout the genome, right? And this is really uncommon. So this turned out to be maybe 30 to 40% of the targets of TDP43 in the cell was piling up on these transposon sequences, right? And this was actually kind of a conservative estimate, because if this transposon happened to lie in the intron of a gene that was expressed in those cells, we gave it to the gene first. So this is a conservative estimate of how many of the binding sites of these things are really falling on transposon sequences. So okay, it binds. How do we know that matters? Well, one of the things that we can do is we can knock out TDP43 in the cells and ask what happens to its transcripts, right? And so if you look at gene transcripts after we knock down TDP43, you'll see some go up, some go down, as you expect when you knock down just about any gene, right? But if you look at transposon transcripts, everything that changed goes up, right? So that suggests that normally TDP43's role is to keep these things silent, is to silence transposons. So it binds, and what's not there binding, levels of these things go up. Okay, and this is true not just in the cell culture model that we were following so that we could do a lot of genomics on this protein, but it's true in a mouse model as well. So when they had neuronal knockout of TDP43 in the mouse, either pan-neuronal or from subsets, you would see that transposon transcripts go up there as well. So basically, anytime you lose TDP43 function, you see that levels of these transcripts are going up. Okay, so this suggests that TDP43 sits here. Maybe it's one of these surveillance proteins that's looking for transposon transcripts, figuring out when it's bound to them, and deciding what to do with those. And in general, it's repressing them, right? But if so, how? Right? So how is this RNA binding protein causing levels of these things to go up when it's not there? How is it normally silencing them? You can think about a lot of different roles for this. You can think that maybe it's literally just a surveillance protein, and it normally figures out and then hands it off to someone else, right? It says, okay, I've got one of these guys. This is not a normal gene transcript. Time to hand this off to the normal transposon control machinery, right? And so that's sort of one model that we have that we're following up. The other idea is that maybe it's actually regulating them directly itself, right? So it's known that one of the roles of TDP43 on gene transcripts is to block translation of those things, and maybe that's one of the roles that it's playing here and putting it through a degradation pathway as a result of that. Okay, so Nick in my lab is following up on these, but I want to tell you something, another story for us that actually involves the Genome Center that I find very cool. Okay, so you're still skeptical. You say, okay, all of these things, transposons or transcripts are going up, in cells and in mouse models, but how do you know this is actually happening in the relevant tissues in a clinical setting, right? In patients with TDP43 aggregates, ALS patients in motor cortex, are we going to see levels of transposons going up as well? And so we started out getting together with a fabulous neurologist at UCSD named John Ravitz. And so he was able to give us a small pilot sample of about 20 samples. 13 of these were from ALS patients, sporadic patients that were known to have TDP43 pathology. And he gave us really nice samples that we were able to get great libraries from. So we got deeply sequenced gene and transposon transcriptomes from these things. And what we saw was maybe a little surprising. It wasn't all of the patients with TDP43 pathology, but it was a reproducible about 25%, right? And that's actually kind of high in ALS, because ALS seems to have a lot of different gene mutations that can contribute to this disorder, and any one of these, no one of these is more than about 20%. Right? So having 25% of them have this signature for transposon transcript elevation was high for this disease. There was one of the controls. We went back in and found out that that was actually a patient receiving radiation who had a brain tumor, and we think we can explain that one that way. Okay, there are also other gene sets that correlate with this. So it isn't just that we went deeper into the transcriptome for those patients, it's that there are other molecular correlates that go along with having elevated levels of transposon transcripts. So whenever we saw ALS patients with very, very high elevated TE levels, we also saw immune pathways going up, right? So things that were in the regulation of the adaptive immunity, glial cell development, oligodendrocyte markers, things like that. And there was another pathway that was actually down in ALS patients, and that was the typical SOD1 oxidative damage pathway. So it looks like if you order the patients by how high levels of desilencing you get, what you're actually doing is you're also ordering them by whether or not they have activation of oxidative damage pathways, which are some of the sort of classical pathways that have been early associated with ALS disease, right? And so here you'll get oxidative phosphorylation. This is actually the KEGG ALS pathway, which is pretty much the SOD1 pathway. So then we actually formed this fantastic collaboration with researchers here at the New York Genome Center who were in the middle of a very large sequencing project to sequence hundreds of patients, whole genome sequencing and RNA sequencing in the same relevant tissues. And they graciously let us get a peek at that data to ask if we saw the same signature there. And we were really kind of surprised and really quite pleased to find that we actually saw a better signature. We saw about half of the ALS patients had the same signature for transposon elevation well above control levels. We almost never saw this in controls in the sequencing data set that we got from them. So this is, again, motor cortex. There's a whole bunch of other tissues. They have so much data that we're still trying to churn through, but we were really quite pleased to see that this replicates in a larger cohort. Okay, so you have one last objection. You're saying, okay, fine, you've got some elevated transposon levels, but lots of things go wrong in dying cells. How do you know this isn't just a byproduct, right, of the thing that's really important, that there's something else important in the cells and this is just something that's happening that correlates with that? Right, so, okay, so we're gonna move to a fly model to test this. So I have a fabulous collaborator. He's a fly neuroscientist named Josh Dubnow. He's now at Stony Brook. He had an awesome graduate student, Lisa Krug, on the project. And they can recapitulate this really quickly in a fly model and test knockdown of lots and lots of other genes, right? So the first thing that I'm showing you here is data just to show you that the fly model actually recapitulates the same hallmarks, right? That when we force aggregation of TDP43 in this fly model, that what we get is we get desilencing of the most abundant endogenous retrovirus in fly called gypsy. So this is the gypsy envelope protein stain in the flies that have TDP43 aggregates. This is a wild type fly of about the same age to show you this isn't just an age thing. And this is it at the RNA level as well. So they get the same signature. Transposons go up. Envelope proteins actually get translated. Okay, and flies with TDP43 aggregates also mimic ALS symptoms in the sense that they also have motor dysfunction. They also have early cell death. So this is a little bit hard to see, but these are tunnel stains showing rampant apoptosis in the fly brain. And they die soon after reaching adulthood. So this is the curve for the flies that have the TDP43 aggregates, and these are the wild type flies, right? And so one of the things we can do is we can say, well, what if we leave the TDP43 aggregates there, but we just fix the transposon problem, right? So what we did is we developed siRNAs against gypsy. That's the most abundant of those endogenous retroviruses that expressed in the fly, and just fixing gypsy levels, not any of the other transposons and not the TDP43 aggregates themselves, actually rescued half the lifespan of these flies. Right? So that suggests at the very least that this is contributing to cell death, and this is contributing to what's actually the problem in this ALS model, right? Regardless of the aggregates and the other things that are going on there, right? So we're really excited about this. But if this is really killing the cells, right? If this is really killing the cells here, then how? Because there's a lot of different ways that transposons can be problematic, and we really need to figure out which one if we're going to actually think about how to fix this, right? So one is that it could just be sort of sequestering the machinery that normally silences these things, and that has other effects on other things, right? There's a double-stranded RNA that's produced whenever these things are expressed, and that could induce an antiviral response, so this could be something like an autoinflammatory disease, right? The envelope proteins that are expressed are typically immunogenic. There have been other studies where in other contexts they've shown that those envelope proteins themselves, just the expression of those is toxic, right? And then of course there's hopping, right? The thing that most people think of when they think of transposon sequences. Okay, and we're still working on that, but at the same time, this is not our work, but work from another lab called Avi Nath. He's a virologist at the NIH, and he's shown that human ALS patients actually do have elevated levels of the human endogenous retrovirus HERV-K that still does make a full-length envelope protein. That envelope protein is toxic in the cells, and the mice that express this HERV-K envelope protein do have much lower survival rates, and he's able to fix that with anti-retroviral retrovirals. So he's really hoping that that will be therapeutic. And we're excited about that as well. Okay, so in summary, your genome is full of transposon parasites. Most of them are non-functional, so I'm not trying to scare you about these things, but some of them can be problematic, and especially when the regulators that normally keep these things silent are compromised, right? And we're just really getting the tools to look at these things more easily with obviously more needed and better reads, better technology needed. Transposons have been linked to numerous human diseases, but it's been sort of scattered here and there without a really general mechanism. And now ALS in at least 2 patient cohorts. More work is needed to find biomarkers early because, of course, all of the samples that we're looking at are autopsy samples at the end stage of the disease, and other genetic correlates that go along with this sort of thing. And while we've linked TDP43 to TE silencing and TEs to neuronal death in flies, we still don't understand how this occurs mechanistically in human cells with a human immune system and whether that's really relevant. So those are the things that we're continuing to work on. And then lastly, I just wanted to thank my lab. So the ones that really work on this are Nick, Regina, Oliver, and Ying, with Wenwei as a former student that was working with both me and Josh Dubnow. I want to say a big thank you to the Genome Center, to Hamali Fatnani's lab and Delphine, who are generating this data, and we're really grateful for them sharing that early. And then we have lots of funding sources. Thank you.